Good afternoon to all the EAPCET aspirants. This is your Prasanna ma'am from NEET WORLD, and yes, we are here with the questionnaire practice of the previous chapter, that is sexual reproduction of flowering plants, and we are here going to start the new unit, that is cell biology, which is a hot spot both for NEET and EAPCET. Let's get started with the questionnaire practice first, and then we will move on to cell the unit of life. So, look at this question beta. This is from pollination, biotic and abiotic pollination. From that area the question is being asked. Just look at this, try to answer. Match the following type of question. Yeah. So, see here, viola, common pansy, commelina and oxalis. These are the plants which are examples for both cleistogamous flowers and chasmogamous flowers. So, A we can take the option as 4. Then abiotic pollination. Abiotic pollination, you can take the example of anemophily, which is the wind pollination. So, B is 2. Next, xenogamy. Xenogamy is nothing but what ma? Transfer of pollen from one flower to stigma of the other flower of same plant or different plant, right? It is to different plant, that is 1. Next, epihydrophily. Epihydrophily example is what? Vallisneria and hydrilla. Vallisneria is one example. So, D you can take the option as 3. Here what is the right set of option? 4, 2, 1 and 3. Option B is the right option. Clear with this? Yeah. Next you look into this one. Identify the character of aileron layer from the following. Where do you find this aileron layer? Aileron layer is the outer layer which acts as an endosperm in the maize seed. Okay, if you can remember the diagram properly, in the maize seed if you can see, the outer kernel is made, the outermost layer is what? Aileron layer. This is proteinaceous. Okay, and inside you will have what ma? Starchy endosperm. Inside you will have starchy endosperm and a papery cotyledon called as scutellum here. Here will be the embryonal axis, where at the tip you will have plumule, at the base you will have radicle. Plumule is covered by what ma? Coleoptile, and radicle is covered by what? Coleorhiza. So, here if you see what, identify the characters of aileron layer. We need to identify the characters of aileron layer only amongst these 4 options. Just look into that carefully and answer beta. Look into that carefully and answer. Try out, no issues. Whether your answer is correct or not at all a problem. Just try out giving the answers. And if you are having any doubt in the option also, you ask me, I will tell you. Okay? Yeah, we have few students here. Please try out answering beta. Yeah. Here we go. First is presence of glycosidic bonds. Glycosidic bonds you can see only in, okay, glycosidic bonds you can see only in the starchy endosperm. So, glycosidic bond is nowhere related to the aileron layer. Yeah, aileron layer is proteinaceous. When it is proteinaceous, it has what ma? Amino acids. Yes or no? It is proteinaceous. So, it is having what? Amino acids. So, B is right. Next, bonds between base and acid forms what? Polymers. Okay. So, if you consider a peptide bond is a bond that is formed in between amino group of one amino acid to carboxyl group of the other amino acid. There a peptide bond is formed. So, that is in between what? Base of one amino acid and carboxyl group, that is acid of the other amino acid. So, this you can consider what, you know, a character for what? Aileron layer for the formation of peptide bond. Next, it separates endosperm with embryo in maize. No, what is separating endosperm from embryo? The papery cotyledon, that is scutellum. Okay, scutellum is separating. Okay, scutellum is separating it from the embryo. So, scutellum is separating the endosperm from embryo. Okay, it is not the aileron layer. So, here what is the correct answer? B, D. Okay, sorry, B, C and D. So, here if you see it carefully, as aileron layer is the outermost layer of this endosperm, this is the thinner layer, right? This also you can consider it as what? A layer which is separating the embryo and the embryonal axis and the endosperm. This also you can consider. Fine. Scutellum and endosperm, aileron layer, both you can consider. Scutellum and aileron layer, both you can consider. Okay. So here that is B, C and D. The option is B, C and D. Okay. Option C. Next. Look at this question about Commelina, whose example is both for chasmogamous and cleistogamous flowers. It consists of both chasmogamous and cleistogamous flowers. Here we go. Commelina produces flowers which open and they are similar to the flowers of the other species, with exposed anthers and stigma. With exposed anthers and stigma are called as what? Chasmogamous flowers. If they are closed, anthers and stigma are closed, so it is cleistogamous flowers. They are called as chasmogamous. A typical angiospermic embryo sac at maturity, it has how many nuclei? 8 nuclei, and it has how many cells? 7 cells. Okay, it has 8 nuclei and 7 cells. So the flowers are chasmogamous flowers. It has 8 nuclei and 7 cells. Clear, right? So this is option D. Next question you look into. Yeah, here. Identify the difference between hercogamy and dicliny in the outbreeding process. So here is a question from outbreeding devices. Okay, from outbreeding devices, if you see, anther and stigma are placed at different positions. Pollen release and stigma receptivity are not synchronized. Production of unisexual flowers, pollen release and stigma receptivity are synchronized. No, they are not synchronized. We need to identify the difference between hercogamy and dicliny. Anther and stigma are placed at different position, that is hercogamy, A. Okay, and production of unisexual flowers is what? Dicliny. These both are the different points. So here we go with A and C, option A. Next you see ma, next question about assertion and reason type of question. Mostly from sexual reproduction of flowering plants you will get assertion reason type of question, match the following, and mostly statement based questions you will get ma. Here you see assertion. In evolution, the distribution of bryophytes and pteridophytes were limited. Distribution of bryophytes and pteridophytes were limited. No, this is wrong. It is not limited. In reason, if you see, the need for water for the transportation of male gamete fertilization limited their distribution. Once again, we will go with the assertion. In evolution, the distribution of bryophytes and pteridophytes are limited, but in comparison with the other divisions of plant kingdom, they are limited only in comparison with other divisions like angiosperms and gymnosperms. Why its distribution is limited in comparative way? We think it is limited because for transportation of male gametes they require what? Water. So here A and R are true, and reason is correct explanation of A, option A. Next, 11th question. Again the same type assertion reason question, just look into it ma. In some plants, the generative cell divides and forms what? 2 male gametes. Try out answering beta, try out answering. In some plants, generative cell divides and forms 2 male gametes during what? During pollen tube growth in stigma. Wrong. During pollen tube growth in style. It is not in stigma, it is in style. The plants which shed pollen in 3-celled stage, the pollen tube will have 2 male gametes from the beginning. True, the pollen tube will have 2 male gametes from the beginning in those pollen grains which are shed at 3-celled stage. So here assertion is wrong and reason is true. Assertion is false and reason is true. Option D is the right answer. Clear, right? Next. 9th question you look into ma. 9th question. Match the following type of question. Animals attracted to flowers by. Animals attracted to flowers by color. Next, beetle flies attracted by what? Foul odor. Flies are attracted by foul odor. Pollination by squirrels is known as what? Therophily. Okay, pollination by squirrels is known as therophily. Hello, who's here, ma? Very good afternoon. Please continue answering the questions, beta. Pollination by snakes is called as ophiophily. Amorphophallus. Amorphophallus, which is by Pronoba moth, this is the safest place. The flowers or the ovary inside is the safest place to lay eggs by Pronoba moth. Fine, right? So here what is the right answer? A is option 3, B is option 4, C is option 5, D is option 1, E is option 2. Fine, right? So here 3, 4, 5, 1 and 2. Option C is the right answer. Next question. Here we go. Look at here. Again, multi-statement based questions. Arrange the sequence of events in microsporogenesis. Questions from microsporogenesis and megasporogenesis from pre-fertilization events are important both for EAPCET exam and a NEET exam. Okay, as such you have to prepare. So see here, beta, first will be what? Pollen mother cell or spore mother cell will undergo the meiotic division. Of course, first meiotic division will happen. After meiotic division, you can see the second thing is what? Microspore tetrad is formed. Okay, microspore tetrad is formed. Post this, okay, dissociation of microspores as pollen grains. Okay, this association of microspore or pollen grains or maturation of microspore to pollen grain is one and the same. This is the third change, and at the time of dispersal of the pollen grains, what happens? Anther will mature and dehydrate. Okay, dehydration which will help in the release of pollen grains from pollen sacs. So this is option 4. So here we go with D, sorry, first is C, then D, and then A, and then B. Sorry. So A and then B. Okay, option C. Right, ma? Next. Identify the correct statements among the following. We need to look at the right statements here. Whenever a statement based question is asked, just look into it whether he is asking you to identify the correct set of statements or incorrect set of statements. Look at here, ability of pistil to recognize the pollen is the result of a dialogue between pollen grain and pistil. Yes or no? Correct. Epicotyle has a short apex and few primordial enclosed in a hollow foliar structure coleoptile. This will give rise to what? Plumule, right? Option C you see. Seed which germinate immediately after formation will be used as store food for the next season. D you look into. In coconut, white kernel is cellular endosperm and liquid is free nuclear endosperm. Option D is correct and C is correct. Epicotyle has a shoot apex, that is true, and it has few primordial cells, but these are not enclosed in the coleoptile. So epicotyle will have few primordial cells. This will mature to what? Plumule. Plumule apex of the plumule is covered by a protective sheet. That protective sheet is called as coleoptile. Fine, right? So here what is the right answer? A, C and D. Option B is the right answer. Okay, ma. Next. Look at this question which is asked in EAPCET 2020. Multi-statement based question again. Multi-statement based question. You can answer this, ma. You can answer. Look at it. Choose the correct statement among the following. We need to choose the correct set of statements. Again, we need to choose correct set of statements. Yeah, here we go. Endothecium with radial thickenings will lose water and contract and help nourishing the developing pollen grains. Okay, yes, this is true. The radial thickenings will lose water. The endothecium radial thickenings will lose water. They will contract and it will help in what? Nourishing the pollen grains. Next, in Helianthus, ovules are covered by single integument. Is this unitegmic? Second, we will look at the all statements once. Production of unisexual flowers will prevent self-pollination in castor. Filiform apparatus will guide entry of pollen tube. Yes, filiform apparatus will guide the entry of pollen tube. D is true. Zoostera, a freshwater grass, shows pollination in water surface. Zoostera will show pollination in water surface. If you see, Zoostera is an example for what? Hypohydrophily. So, E will be wrong. When E is wrong, you can eliminate these two. Find right. Yeah. Option B and C you can eliminate. You look at this beta, B in Helianthus, of course, it is unitegmic. It has single integument, as the petals are gamopetalae here. They are fused, right? And next here, if you see option C, production of unisexual flowers, of course, they will prevent self-pollination in castor. This comes under what? Diclining type of outbreeding device. So, here B, C, D will be correct. Option D is the right answer. Okay. Any doubt anywhere, ma? Yeah. Next question. Look at here. This diagram I gave you for a reference. Don't look at here this now. Just try identifying the sequence first. Try out identifying the sequence. Try out identifying the sequence. What is the correct arrangement of the following parts from the lower end in the monocot embryo sac? From the lower end we have to see. From the lower end of monocot embryo sac. Diagram based question. Okay, the question is diagram based. Here we go. First, from the base to tip you see, ma. At the base we have what? Coleorhiza. Coleorhiza, above coleorhiza we have what? Root cap. Above root cap we have what? Radicle. And here is the epiblast. Epiblast is nothing but the remnants of the second cotyledon. These are remnants of second cotyledon. You can also say this is the underdeveloped second cotyledon. Okay, next above this you have shoot apex, coleoptile, lastly it is scutellum. So, now you start organizing this in sequence with a first is what? Coleorhiza, second will be what? Root cap, coleorhiza, root cap, above which you have radicle, above which you have epiblast, above which shoot apex, above shoot apex you have coleoptile, above coleoptile you have scutellum. So, here we go with the right set. First is what? From E followed by G, followed by A, followed by D, then C, F, B. Option C is the right answer. Clear? Everybody got this question? Any doubt anywhere? Next, this one. Identify a typical angiospermic embryo sac from the following. Of course, we know the angiospermic embryo sac will have both ends, micropylar end and chelazal end, and it will have how many nuclei? 8 nuclei. 3 cells at the chelazal end, 3 cells at the micropylar end, which is egg apparatus, and 2 nuclei at the center, combinedly called as what? Polar nuclei or central cell. Okay. So, it is completely how much ma? 8 nucleated 7 celled condition. See, on the angiospermic embryo sac, this is the second question we are seeing from the previous year questions. That important is the embryo sac structure. Just look into it, ma. Next, 18th question, look into it carefully. Arrange the following based on the number in ascending order. Based on the number. Very interesting question actually. Just go through it and answer. You can do it. First, you see the number of haploid cells present in a flower. Haploid cells he is asking. Haploid cells present in a flower with 121 megaspore mother cells. Okay. Development on maturity. 121 megaspore mother cells. After undergoing reductional division, of course, each megaspore mother cell will give rise to 4 megaspores, of which at maturity he is asking. At maturity 3 will be degenerated and only 1 will be functional. In that case, 121 megaspore mother cells, how many haploid cells will be there at maturity? 121 megaspores will be there. You can call it as functional megaspores. Functional megaspores at maturity. That's it. Simple, right? Next, you see number of pollen grains released in a flower showing anemophily. Numerous pollen grains. There is no exact number. Okay. Anther lobes in hibiscus. Hibiscus being polypetalous. Okay. Hibiscus is having how many lobes he is saying? Anther lobes in hibiscus is actually anther here in hibiscus is monothecous anther. Monothecous means single theca or single lobe, right? Monothecous which has single lobe. Next you go through number of integuments in the ovule of polypetalae. Polypetalous ovule is having what? How many integuments? It is bitegmic. Okay, it is bitegmic. Bitegmic means it is having 2 integuments. Fine, right? It will be having 2 integuments. Next, number of primary endosperm nucleus chromosome number in maize flower with 48 meiotic divisions. With 48 meiotic divisions during megasporogenesis. Okay. Number of primary endospermic nucleus chromosome number in maize flower with 48 meiotic divisions. Each meiotic division, just a second, primary endospermic nucleus chromosome number in maize flower with 48 meiotic divisions in megasporogenesis. 48 meiotic divisions in megasporogenesis will give out 48 functional megaspores. Yes or no? Functional megaspores. Each megaspore will provide 1 embryo sac. So, 48 functional megaspores will give out 48 embryo sacs. Okay. Each embryo sac will have one primary endospermic nucleus. Okay. As such, 48 will be the chromosome number. Now, you start putting it in the ascending order. Anther lobes are 1. Okay. So, first will be option C. Okay. Anther lobes are 1. Then followed by number of integuments, 2 integuments, right? Here in bitegmic for polypetalae ovule. Okay. Ovule will have 2 integuments here. So, C will have only 1 followed by D is having 2, the number is 2. So, C, D followed by what ma? A, it is having how many megaspore mother cells? 121. So, 121 functional cells will be there. Just a second. 2 minutes ma, work on this question. Yeah, so it will be first B is what ma? 1 followed by C, sorry, first C which has only 1 theca or 1 lobe, followed by D which is polypetalae and it is having 2 integuments. Okay, so it is C, D and then A followed by E and B, okay, which has numerous or infinite pollen grains. So, here the right option will be option B. Any doubt here from this question ma? Any doubt here from this question? Right. Next, choose the correct statement. Right set of statements we have to choose. Several hormonal and structural changes are initiated for the development of floral primordia. Yes, this is true. For the development of floral primordia, many hormonal and structural changes will happen for the development of floral primordia. Each anther will have 2 sporangiate sites. Wrong, it will have how many? 4 sporangiate sites. Stomium will be useful for the dehiscence of pollen sacs. True. Pollen grains can be stored at -196 degrees Celsius. True. Pollen grains viability in rice is 2 to 3 days. Wrong, it is for months, right. So, here pollen grain viability will be for months in Leguminosae rosaceae families. The viability is for few days in rice and cereals, right. So, E is true. So, here what options we can opt? A, C and D. A is true. Each anther will have 4 sporangiate sites. So, here look into the options A, C and E, right. Next question, what is apomixis? The question is on apomixis. Production of seeds without fertilization, and the seeds are addressed as what? Apomictic seeds. Seeds are addressed as apomictic seeds. Option D. Next, assertion reason type of question. In assertion you see ma, in angiosperms the outer layer exine of pollen grain is made up of what? Sporopollenin. The inner intine layer carries 2 male gametes to vicinity of the egg cell and the central cell of the female gametophyte. Female gametophyte here is what ma? Embryo sac. Fine, right. So, here assertion and reason both are true, but nowhere it is the correct explanation, nowhere it is explaining about the sporopollenin composition in the pollen grain. There are 2 distinct unique points. So, here the option will be B. Next question you look into assertion and reason type again. Assertion and reason type again. Yeah, in assertion if you see removal of anther from bisexual flower before anther dehiscence is called as emasculation. Okay, true. Emasculation prevents the self-pollination in bisexual flowers. Correct. So, here reason is explaining assertion. Why emasculation is being explained in the reason. So, here A and R are true. R is the correct explanation of assertion. Option A is the right answer. Next question about match the following type. This is from types of biotic pollinating agents. Question is on types of biotic pollinating agents. Entomophily is the pollination by insects. Chiropterophily, pollination by bats. Thyrophily, pollination is by squirrels. Ophiophily, pollination is by snakes. Okay. So, here we go through first entomophily, that is option 3, B is option 5, C is option 1 and D is option 2. B is the right answer. This you see. A typical angiosperm embryo sac at maturity. At maturity, typical angiospermic embryo sac will be having how many nucleus? 8 nucleated 7-celled condition. Option B. 8 nucleated 7-celled condition. 25th you see ma. In which of the following plants hydrophily is common phenomena? Hydrophily is common phenomena in hydrilla here. Water lily, water hyacinth, they do not have hydrophily. Amorphophallus, the pollination is not by water. Hydrilla, Vallisneria will follow epihydrophily, Zoostera, hypohydrophily. Clear, right? So, the right answer is option C. Next question, assertion reason type again. This is also on the types of pollination. On types of pollination you got most of the questions, not even in the EAPCET but also in the NEET exam. As such, have an additional focus and interest when you are going through the concept of types of pollination, both biotic and abiotic pollination. Just look into it and answer ma. Assertion. Xenogamy involves transfer of pollen grains. Transfer of pollen grains from the flower of one plant to the stigma of another plant. True. In reason, this is the only type of pollination which brings genetically different types of pollen grains to the stigma. True. So, in reason it is explaining about xenogamy, that is cross pollination. So, both A and R are true and reason is correct explanation of assertion. Whenever you have to choose between whether it is correct explanation or not, look into it whether it is explaining why, how, what is the process that is dealt in assertion. If it is explaining any of the questions that you have put over there like why, how, what, then you can go through that it is the correct explanation. If at all how many questions you have put after assertion also, in reason no way it is explaining assertion, then you have to choose option B. But put as many questions as possible like is it explaining, how is it explaining, why is it explaining, what is it explaining. Based on that only you can choose whether it is R is the right explanation or not. Okay. Next. Simple question, this is from the dicot embryo development. dicot embryo development. Yeah, dicot embryo development. First you see ma, pro-embryo, globular embryo, mature embryo and heart-shaped embryo. No. Mature embryo will be at the end. First heart-shaped embryo will not come before globular embryo. No. Mature embryo will not come at the beginning. So here you see, first the sequence stages of the embryogenesis. First will be pro-embryo, followed by globular embryo, followed by heart-shaped embryo, lastly mature embryo, which is option D. So 27, the right answer is option D. Next you look at this question. Read the following statements. Statement based question again. Pollen grains contain a thin outer layer that is exine, which is made up of sporopollenin. True. This is made up of sporopollenin. Sporopollenin is an organic material which is resistant to high temperature and strong acids. Yes, this is resistant to high temperatures and strong acids. Pollen grains are preserved as fossils. Yes, they are preserved as fossils. Consumption of pollen products does not increase performance. No, they will increase the performance of the athletes. Okay, so here 1, 2, 3. 3 options are correct, ma. 1, 2 and 3. Option A you can choose. 2 and 3 also correct. 1 and 3 also correct. Okay. Yeah, next. We will go with the next question. This one. Again statement based question. Look at the question properly. Here he is asking you to identify false statements, means incorrect statements. Look into it carefully and answer. Symbol. Endosperm is triploid, filled with reserved food, and this will nourish the developing embryo. Yes, endosperm is triploid. Okay, this is filled with reserved food material and this will nourish the developing embryo. Formation of pollen grains from megaspore mother cell. Formation of pollen grains from megaspore mother cell is called as what? Megasporogenesis. True. 2 nuclei are formed from the functional megaspore. Yes, 2 nuclei are formed from functional megaspore by what? Mitotic divisions. Wrong. And moves to the opposite poles of the embryo sac. By mitotic division from the functional megaspore, 8 nuclei are formed, not 2 nuclei. At maturity, a typical embryo sac has 7 nuclei. No, it has 8 nuclei and 7 celled condition. So here the right answer is 1 and 2. Okay. Yeah, 1 and 2. Option C. Okay. Next. Last question from sexual reproduction of flowering plants. Assertion and reason type. Just go through it better. Cellular thickenings at micropylar tip will guide pollen tubes into the synergids. Yes, cellular thickenings at micropylar tip, they will guide the pollen tubes into the synergids. True. Synergids have antipodals located at the chalazial end. Wrong. Synergids will never have antipodals. Antipodals are the vegetative cells which are located at the chalazial end. Antipodals are vegetative cells located at the chalazial end. So here A is true and R is false. Option C is the right answer. Clear, right, ma? Any doubt anywhere? Yeah. Fine. Now we will move on to the chapter Cell the Unit of Life. From this chapter Cell the Unit of Life, we can expect 1 or 2 questions in your EAPCET examination, or 2 questions, 2 to 3 questions you can expect in your NEET exam as well. And the content is same with respect to this chapter for both EAPCET and NEET. How you have to learn this chapter in detail? Okay, Cell the Unit of Life. First, let us look into the framework of this chapter. Okay. Cell the Unit of Life. Look into the framework of this chapter. First, from Cell the Unit of Life, you have to fragment this chapter into 3 areas. Okay. First is on the discoveries of cell. Okay. Cell discoveries and different cells based on size and shape. Later, which you have to learn about cell theory, which is very important. Cell theory followed by its exceptions. Okay, which is very important. And then you will move on to prokaryotic cell, and eukaryotic cell. Fine, right now, prokaryotic cell and eukaryotic cell. Here from prokaryotic cell, if you see, prokaryotic cell, complete structure and its description you have to learn, and whenever you are revising prokaryotic cell, revise it along with kingdom Monera from biological classification. Kingdom Monera from biological classification. Fine, right? Now we will move on to eukaryotic cell. From eukaryotic cell, the very first thing you have to do is to learn about the cell wall, outer boundaries, which is cell wall from plant cell, and from animal cell it is cell membrane. Both the structures you have to go through, and when you are going through the cell membrane, you have to be very clear on the note about fluid mosaic model. Okay, you have to be very clear on fluid mosaic model. Okay, so here we go about plant cell and animal cell. From plant cell you will learn first what? Cell wall. Here cell membrane. In cell membrane, what is the major model you will look into, beta? Fluid mosaic model. Fine, right? Okay, and then after which, and then you will go through from here non-membrane bound organelles, single membrane bound and double membrane bound organelles, both from plant cell and animal cell. Okay. First is non-membrane bound. Fine, right now. Non-membrane bound organelles, the things you have to exercise over here is ribosomes, of course, and centrioles. Very important, centrioles again. Single membrane bound, you have to go through the major system, that is endomembrane system here. Endomembrane system. Fine, right. Next is double membrane bound organelles. Here you look at the double membrane bound organelles. First is nucleus. When you are going through nucleus, you have to be clear on the structure of chromosome. And shapes of the chromosome. Fine, right. After which you have to go through plastids. 3 types of plastids again, which includes chloroplast, chromoplast, followed by leucoplast. Fine, right beta. Lastly, it is mitochondria. This is the complete framework of this chapter. No question will escape from the contents that we have put over here in this single slide, either from your EAPCET or from your NEET exam. Okay. So, this should be a flow of preparation beta. First, when you start out with this chapter, look into the cell discoveries, okay, and then different shapes of the cells. Okay. Yeah, then what ma? Different shapes of the cells and different cells based on the size. After which cell theory and its exceptions. And prokaryotic cell, its complete structure and movement, everything you have to, types of reproduction, types of nutrition, everything you have to go through. And if you are at your revision pace, please revise the prokaryotic cell along with the kingdom Monera from biological classification. Questions will asked in linked between these two concepts of 2 chapters. Next, when you are going through prokaryotic cell, please highlight on the gram staining as well. Okay, go through gram staining as well. Next is eukaryotic cell. You have to pick out both plant cell and animal cell. Plant cell, first finish off the cell wall, and different compositions of cell walls and different cells. Okay, like fungal cell wall composition, algal cell wall composition, bacterial cell wall composition, please go through here. And then from animal cell, you have to work out on the fluid mosaic model, which is giving out the clear note on the cell membrane. After which you will look into the cell organelles. Cell organelles, single membrane bound, non-membrane bound and double membrane bound. Non-membrane bound includes ribosomes and centrioles. Single membrane bound, where you have to learn completely about endomembrane system. Here 4 organelles will come into picture. What are those 4 organelles beta? Endoplasmic reticulum, Golgi bodies, and then vacuole, and then lysosomes. Okay, these are the 4 organelles that you have to go through. And then double membrane bound organelles, first nucleus, when you are going through nucleus, about structure of chromosomes and shapes of chromosomes. Then double membrane bound organelle plastids, you have to go through chloroplast, chromoplast and leucoplast. Lastly, it is mitochondria. These are the double membrane bound organelles, and this is the complete framework of this chapter. High hotspots of this chapter you can go through is first cell organelle, cell theory, one hotspot. Then from kingdom Monera, here you can go through. Endomembrane system is the major hotspot. structure of chromosome, shapes of the chromosome, another hotspot, and then fluid mosaic model is the major hotspots. These are the hotspots of this chapter. From tomorrow we are going to begin with this chapter, followed by EAPCET previous year questionnaire practice. So don't miss out this. This is useful for all the students who are working out for your 2026 examination. Please be there. At least watch the recorded sessions. It will be useful for toughest topics. And look into the EAPCET P/Q questionnaire practice, which is covering the concept as well. Okay. So that's all for today's session, students. Thank you. Just go through the concepts that are given over here. Okay, fine. If you are already fine-tuned with this chapter, just go through these hotspots, which is clearly analyzed both from your EAPCET and NEET PYQs. Okay, yeah, that's all for today's session now.